It leads naturally, that idea, the cloning idea, leads naturally, certainly when you look back, to Shinius of the experiment, where he showed that the introduction of just 4 carefully selected transcription factors into cells is capable of changing a small proportion of them to the cell type from which you've taken the transcription factors, in this case, induced pluripotent cell. But there are a number of strange things about this procedure. I mean, it's wonderful. I think its experiment is certainly one of the most important of this past decade. May very well be one of the most important of this century. So please don't misunderstand me. But it does have a number of edges to it, a number of loose ends. Some of them are a little bit different from the ones which are usually mentioned, like using retroviruses which have an integration site is a significant disadvantage. There's variation in the expression of them. These sort of things is often mentioned. I don't think some of these things are not usually mentioned. Partial reprogramming, I think, is mentioned. That you get cells which begin to go down this process, but don't quite go all the way to reprogramming. But a number of groups have noticed that during the early phases of this production of induced pluripotent cells, there's a whole zoo of different cell types there. And if you pick them out carefully, you can actually isolate cells which have a different phenotype. So Mickey Batch's lab in Canada picked out multipotent hematopoietic cells from that pool and characterized them fairly well. As well as using the full range of the 4 young marker factors, he also showed that the same thing could be achieved if you only introduced OCT4. So the sequence is that you put in the transcription factors and then you change the cells to the medium which you think is appropriate for that particular cell type you're picking out. So as these multipotent cells were picked out and they could give rise to different cell lineages, but not the lymphoid lineage. So these are not true hematopoietic stem cells, they're multipotent cell type. He showed that they were capable of engraftment in vivo. He also showed that they had not gone through a pluripotent stage on the way to getting there. This is a direct hop from a fibrous population to a hematopoietic progenitor. We did the same thing, which is not published, picking out neural stem cell progenitors from the same mix, and found, as he did, that OCT4 was capable of doing it on its own. There was an increase in efficiency and speed of the process if you put MIC in as well, but OCT4 was the only one which could do it all on its own. We did not show that the cells could function physiologically. We grew them out, maintained them in culture for a long time, and got different neural lineages, but did not take it any further than that. So there's something slightly strange going on. The situation isn't being controlled quite as accurately as you might hope in this early stage of reprogramming. No doubt lots of people began to think, well, if this will work, taking fibroblasts and going to pluripotent cells, can we find the factors which will enable us to go to other cell types? And many of you probably know that there are at least 2 papers, the references are cited briefly on the slide, where people have changed fibroblasts into either neural population or into cardiomyocytes in the mouse. With a low efficiency, but they did manage to get the cells to go to a different phenotype. Other papers. I'm not quite sure why this hasn't been pursued more actively, because there seems to me to be real potential value in thinking about not going to the pluripotent cell stage, but going to a progenitor cell stage. The other disadvantage of this approach is that you must have identified at least enough of the key factors in order to be able to reprogram cells in this way. It's slow. You don't get colonies for several weeks. It's inefficient and it's inaccurate. A key skill, my colleagues would tell me, in making IPS cells is in learning to pick the right colonies. I think it would be a good indicator that a new method has arrived, which is more accurate and in some ways perhaps more useful, if it was quicker, if the efficiency was higher, but also if you only got the cell type that you were looking for, rather than getting this rather confused state. So this is one challenge for those of us who work on changing cell fate. The other thing I've touched on already is to wonder whether we spend too much time looking at pluripotent cells. That leads me into the last few slides, which relate to stem cells. I do agree with the remark that's been made several times that the first use of cells is likely to be in identifying drugs. But of the people who've spoken here, I think I'm probably the most optimistic in terms of moving towards cell therapy. Not next year, but maybe 10 years, 5-10 years away, getting into serious clinical trial. These are some of the requirements that we would have if we're thinking of cell therapy. Ideally, we want immunologically matched cells. Critically, we want cells of the correct tissue. And there mustn't be contamination by other cell types. In particular, we're very anxious about having pluripotent cells present in the mix. I've no doubt that one of the reasons why Geron and other companies spent a long time preparing data for the FDA is because they were having to convince them that there were no or very few pluripotent cells in the mix. I guess you all know, as I do, that they put in more than 20,000 pages into the FDA. Now, if you were using a cell population which was never pluripotent, if you could take the cell type which is convenient to work with from the patient and turn it directly into the population that you want, and perhaps a progenitor population, you would have a different risk, a lesser risk, but a different risk. Of course, you do need to have very large numbers of cells, which would be one strong reason, I think, for thinking of working with progenitors rather than terminally differentiated cell. So this is the same diagram that I used previously, but just putting on, in addition to this list of the two that I know about, the suggestion of working with progenitors. Lots of people have thought about working with progenitors in different contexts. Lots of people have mentioned them in posters outside there in the hall. It would be very interesting to see whether it's possible to take the blood cell or the skin cell or whatever it is from the potential patient and turn them into a progenitor from the tissue that you're interested in by some form of direct reprogramming. See, I think that I've described to you what I see as a trajectory. We thought, let's say when I was a boy, it's not that long ago, that things were fixed. And then we discovered that, well, maybe they're actually not quite as well fixed as we thought. You can change them. The oocyte's really quite good at it. And then we got to be better at fixing them by manipulating them more precisely with particular proteins. And we could go to pluripotent cells. And quite genuinely, we thought that was magical. But why have we stopped thinking about the possibility of going to different cell types which might meet other needs? Pluripotent cells will be useful sometimes if you're wanting more than one cell type. But if you're thinking of RPE cells, for example, why not produce them directly, rather than going through that pluripotent mix? So I personally believe that our ability to change fate accurately will become much more routine than it is at the present time. Procedures will become simplified because we'll get to know what we're doing and do it more accurately and effectively. So the accuracy of the change will be improved so that we won't have that zoo of cells at the beginning of the process, but we'll go straight to what we want. And so these cells will offer a great deal because they'll not only be useful to study inherited disease, but they'll begin to move us in the direction of cell therapy. Now, there may be potential patients or patient advocates in this room, so can I emphasize this isn't next week, this may be 10 years away. I recognize that a significant proportion of this audience is young people who are early in their stage of research. So I think they'll definitely live to see this. I hope I will. I actually think I will, but I might be wrong about that one. But I think that there's a balance there I think we should be excited by this sort of possibility. We should work towards it. But we do have to be realistic when we're talking to people who might depend on this for improved health. It will take 10, 20 years at least before it gets into routine therapy.